Good day, everyone. This is Martin Akerman speaking. I'm the co-founder and CTO of Envisagenics. Today, I will discuss an important topic around the use of AI for drug discovery, and this is how to take AI-driven computational predictions to the laboratory and subject them to experimental validations to verify whether they are real or not. I want to make the following point today, that if AI algorithms are more understandable, relatable, or transparent to partners, scientists, or key decision makers in drug development, it is more likely that they will trust them enough in order to spend time, money, and efforts in performing those lengthy validations necessary to move forward with AI-driven targets. So I'd like to open up with the unfamous and problematic black box effect of AI. The black box effect is another way of saying we don't fully understand or know the reason why a given machine learning algorithm is working. It is entirely possible that the machine learning will make the right prediction, but in often cases, we don't know what drove the algorithm or how the algorithm arrived to a given prediction. In fact, the biggest problem of the black box effect is what happens downstream after the prediction. What to do with those predictions once we have them? Should we trust them? How should we act upon them? So one interesting idea here is that the longer it takes and the harder it is to verify a machine learning prediction, the more problematic the black box effect becomes. Some predictions are easier to verify than others. For example, if we are using AI for image recognition, it often takes shorter to verify an image. A pathologist looking at suspect melanoma can do that but by naked eye. But when it comes to drug discovery or target ID, things are different because an algorithm can predict a new drug target and say, hey, this is the next cure for cancer. However, it will take from months to years and a lot of resources to validate that findings. So scientists naturally will ask, how do you know that prediction is correct? So what I'd like to propose today is that there is a way to open the black box, or making it grayer for a different analogy. But let's stick with opening the black box. And opening the black box means to help people understand machine learning predictions and compel them to act upon them. And this can be done by building a more transparent and interpretable AI with using predictive features that people can understand and relate to. So the next question is, what goes into this more transparent AI model? And I will count three things. One, the biology. The model needs to be a hypothesis-driven kind of a model. The more specific the biological questions that we ask, the better. For example, we can build a classifier to predict the difference between patients and a control, but we have to put that in a context. Like, for example, due to a given drug treatment, we can't just expect to find difference between two groups without a context. Second, the domain knowledge. Many of the questions that we ask have been investigated before, so if there is a way to incorporate that prior knowledge into the model, that would be great. For example, if we are predicting drug response or any mechanistic information, if we have any mechanistic information that we trust about that drug from the literature or anything else, we can incorporate that into the model, and that can certainly help. And finally, using intuitive information, that is to use discrete, quantifiable information. For example, in image recognition, people use the continuum of pixels information as training material. In drug discovery, we don't really recommend to do that. My rule of thumb, at least at Envisagenics, is that if you cannot articulate in English what the feature is measuring, then don't use it. And we think following these rules, we can get great, transparent, predictive models. So today, I'll walk you through a proof of concept from my company, Envisagenics. Our focus is target identification using machine learning, cloud computing, and our bread and butter, that is RNA-seq data. We are experts in RNA biology, particularly in splicing regulation. So our premise is to identify disease-specific aberrant splicing that can be targeted with different modalities, including antisense, antibodies, and small molecules. So in line with this discussion, our biology is target identification, our domain knowledge is RNA splicing, and our intuitive information is derived from RNA sequencing data. So here we have an overview of the SpliceCore platform. SpliceCore takes as an input RNA-seq data and produces drug targets for different modalities, including splicing switching, antisense, oligonucleotides, and protein isoforms that we can target with antibodies or small molecules. So the incoming RNA-seq is first assembled and processed using our scalable cloud on Microsoft Azure, and then it connects with a number of machine learning algorithms that interrogate the data with specific biological questions. For example, one of the models here on top, it's called SpliceLearn, and it predicts druggable targets for antisense molecules that modulate alternative splicing. Today, I will focus on experimental validations for SpliceLearn, this algorithm for antisense drug discovery that will be our biology in this story. But first, let me tell you about our domain knowledge, splicing. So more than 99% of human genes undergo splicing to obtain mature mRNA molecules. Without splicing, mRNA cannot mature and cannot be translated, so this is a very important process. Splicing is regulated by a macromolecule called the spliceosome, which is also the biggest and most complicated molecular complex in human cells. It has over 300 proteins that are assembly into subcomplexes and moving parts. So now the thing about a big complex is that it has many vulnerabilities, many moving parts, so many things that can go wrong and cause disease in the spliceosome. So spliceosomal mutations and errors are known to cause 370 diseases, and there could be others, but some scientists call these diseases by the name of spliceopathies. Now, when the spliceosome is mutated, it leads to widespread splicing errors and generation of disease-specific splicing isoform. So in cancer, for example, there are many well-known examples in the literature of splicing events that affect multiple hallmarks of cancer, like proliferation, mesenchymal transition, angiogenesis, others. So what SpliceCore does is to identify splicing events of this sort. It predicts both the target and the antisense compound necessary for the splicing modulation. So today I want to show you one of the proof of concept programs that we have in triple negative breast cancer, or TNBC for short. Here is our pipeline, 100% novel targets. And as you can see, some of them are targetable by different modalities, including antibodies and small molecules as well. So since our laboratory in-house specializes on antisense, we are actively seeking for partners to co-develop antibodies for targets such as ENV21. But today, I will focus on ENV2. This is an antisense target. It's a novel target for TNBC. And to tell you a little bit about the target that we found, we analyzed over 1,300 RNA-seq samples from triple-negative breast cancer from public and proprietary sources. We identified over 20,000 alternative splicing events. One of them we call ENV2, and it was an exon skipping event observed specifically in triple-negative breast cancer. We've seen that 60.5% of the patients we analyzed presented this exon skipping event, and it was also associated with poor survival outcome. As you can see, ENV2 is differentially spliced in basal epithelium, but not other breast tissues, and the effect is only seen at the alternative splicing level, but not at the gene expression level. That means that using conventional tools for gene expression analysis, we would have missed this target. Here we can see the splicer profile of ENV2. The purple line indicates the probability that delivering antisense to a given nucleotide position that would elicit a splicing change of the proximal exon. The red lines are the computationally selected binding sites for antisense, and the pink in the bottom are the actual antisense that we synthesize at the lab. And here you can see the experimental validation. As predicted, the compound ENV205 modulates alternative splicing of ENV2, and it also, as you can see on the right, it kills the cancer cells specifically in basal epithelium, but not luminal cells. ENV2 is a novel target for triple-negative breast cancers with no record in the literature whatsoever. We're investigating ENV2 in our laboratory and discovering that it has very interesting biology. Here we can see evidence that ENV2 modulates the TGF-beta pathway, which is important for tumor progression. The graph on the left shows TGF-beta dependent upregulation of several TGF-beta downstream targets, including P21, in a 3 hours TGF-beta treatment in MB231 cells. On the right, we see the reversal of the baseline of P21 levels upon addition of the antisense, again, the 0502, that promotes the splicing switching, which is not seen in control oligos. So I'm running out of time, so I want to say before I go is that all this work from RNA-seq data analysis to compound, it took us only 8 months, thanks to the use of AI. And finally, that we are active in other areas beyond TNBC. We're working in other areas of oncology, as well as NASH, NFLD, and neurodegeneration, and actively looking for partners which are interested to use our approach and work together in developing therapeutics in those areas. So if you stuck with me all the way to the end, thank you so much for listening, and I'm happy to somehow take questions through this interface. Thank you.